In the vast and varied kingdom of birds, life is a constant dance of survival. From the smallest hummingbird to the grandest eagle, each creature faces its own unique challenges. Yet sometimes the greatest threats are those unseen microscopic adversaries that can sweep through a population with devastating speed. One such peril is fowl cholera. This disease, known to scientists and bird keepers alike, represents a significant concern for avian health across the globe. It is a spectre that haunts domestic poultry farms and can cast a shadow over wild bird populations, a silent disruptor of the natural order for many feathered species. Fowl cholera is not a new affliction. Its presence has been documented for centuries, a persistent problem for those who raise birds for food or companionship, and a natural check on wild populations. The illness is caused by a bacterium, a tiny single-celled organism named. This microbe, though minuscule, possesses the power to cause widespread illness and mortality. Understanding fowl cholera is crucial, not only for the welfare of domesticated birds, but also for the conservation of wild avian biodiversity. Its impact resonates through ecosystems and economies, highlighting the delicate balance that exists within the natural world and our interactions with it. The importance of comprehending this disease cannot be overstated. For poultry industries, an outbreak can mean catastrophic economic losses, affecting livelihoods and food supply chains. For wild birds, particularly those in dense colonies or those already stressed by environmental changes, fowl cholera can lead to significant population declines. It can tip the scales for endangered species, pushing them closer to the brink. Therefore, knowledge about its symptoms, how it spreads, and how it can be controlled is paramount. This understanding empowers us to protect these remarkable creatures, from the humble chicken in a farmyard to the magnificent waterfowl in their wild wetland homes. This exploration will journey into the world of fowl cholera. We will look closely at the bacterium itself, the ways it infiltrates avian communities, and the signs it leaves in its wake. We will examine the methods used to identify this stealthy invader and the strategies employed to shield birds from its grasp. The story of fowl cholera is a reminder of the intricate connections that bind all living things and the constant vigilance required to maintain health and harmony in the animal kingdom. It is a tale of challenge, but also one of hope, as science and careful management strive to mitigate its impact. At the heart of fowl cholera lies a microscopic organism, the bacterium. This tiny life form, invisible to our unaided eyes, is the sole architect of this often devastating disease. It is a gram-negative bacterium, a classification that refers to how it reacts to a specific staining process used in laboratories, providing clues about its cell wall structure. It is not a single uniform entity, rather, it exists in several different strains or serotypes. Some of these strains are relatively harmless, residing in the respiratory tracts of healthy animals without causing illness. Others, however, are highly virulent, capable of causing severe disease and rapid death in susceptible birds. The bacterium is remarkably adaptable. It can survive for a period in contaminated environments, particularly in damp soil, water sources, or organic matter like bird droppings and carcasses. This resilience allows it to persist and await a new host. It's not exclusively a bird pathogen. It can also affect a wide range of mammals, including humans, though fowl cholera specifically refers to the disease it causes in birds. The specific strains that cause severe disease in birds are often highly adapted to their avian hosts, making them particularly efficient at causing infection within these populations once introduced. The virulence of strains can vary significantly. Some strains might cause only mild localized infections, while others can lead to a systemic infection spreading throughout the bird's body via the bloodstream, resulting in a condition known as septicemia. The bacterium possesses various tools or virulence factors that help it to invade the host, evade the immune system, and cause damage. These can include a capsule that protects it from being engulfed by immune cells and toxins that damage host tissues. It is this arsenal that makes certain strains so formidable. Understanding the nature of is the first critical step in combating fowl cholera. Knowing its different strains, its ability to survive in the environment, and the mechanisms by which it causes disease informs every aspect of prevention and control. It allows scientists to develop effective vaccines, guide sanitation practices on farms, and helps wildlife managers to understand disease dynamics in wild populations. This tiny organism, though unseen, commands our attention due to the profound impact it can have on the avian world, a world we are so intrinsically linked to. When fowl cholera strikes in its most aggressive guise, it is known as the acute form. This manifestation of the disease is characterized by its sudden onset and rapid progression, often leading to death within a remarkably short time frame. Affected birds may appear perfectly healthy just hours before succumbing. This swiftness can be alarming for bird keepers, as a seemingly thriving flock can be decimated almost overnight. The first sign might simply be finding dead birds with no prior warning of illness. This sudden mortality is a hallmark of acute fowl cholera, a testament to the potent virulence of certain strains. For those birds observed before death, the symptoms of acute fowl cholera can include a high fever, making the birds feel hot to the touch. They often exhibit extreme lethargy and depression, appearing listless and unwilling to move or eat. Ruffled feathers are a common sight, as the bird no longer has the energy or inclination for preening. Labored breathing, sometimes with a gurgling sound, may occur as the infection overwhelms the respiratory system. A watery, greenish or yellowish diarrhea is also frequently observed, leading to dehydration and further weakening of the bird. These signs paint a picture of a body rapidly overwhelmed by infection. Internally, the damage caused by acute fowl cholera is extensive. Post-mortem examinations, known as necropsies, often reveal widespread hemorrhages or bleeding in various organs. The liver may be enlarged and contain small pinpoint areas of dead tissue, known as necrotic foci. The heart, too, can show signs of hemorrhage on its surface. The lungs are often congested and filled with fluid, explaining the respiratory distress. This systemic infection, where the bacteria multiply rapidly in the bloodstream and invade multiple organs, is what leads to the swift demise of the bird. It is a brutal and efficient assault. The sheer speed of acute fowl cholera makes it particularly challenging to manage once an outbreak begins. There is little time for treatment to take effect, and the priority often shifts to preventing further spread within the flock and to other susceptible groups. The sudden loss of multiple birds is a distressing experience, underscoring the severe nature of this disease. It serves as a stark reminder of the vulnerability of even the most robust-seeming creatures to the power of microscopic pathogens that can invade and conquer with terrifying efficiency. While acute fowl cholera brings swift devastation, the disease can also take a more insidious, lingering form known as chronic fowl cholera. This occurs when birds survive the initial acute phase of infection or are infected with less virulent strains of. Instead of rapid death, these birds endure a prolonged illness, often with localized infections that can persist for weeks or even months. The chronic form, though less immediately fatal, still causes significant suffering and can lead to debilitating conditions, impacting the bird's quality of life and productivity in a flock setting. The symptoms of chronic fowl cholera are varied and depend on which parts of the bird's body are affected. One common manifestation is localized swelling, often in the joints of the legs or wings, leading to lameness and difficulty moving. Swellings can also occur in the wattles, those fleshy appendages on the heads of some bird species, which may become enlarged, hot and tender. These swollen areas are typically filled with a cheesy, pus-like material, a result of the bird's immune system battling the persistent bacterial infection. Such infections can cause considerable pain and discomfort. Respiratory problems can also persist in the chronic form. Birds might exhibit a persistent cough, sneezing, or a nasal discharge. Sometimes, the infection settles in the sinuses, causing swelling around the eyes. In other cases, the middle ear or the cranial bones can become infected, leading to neurological signs such as twisted necks, a condition known as torticollis, or a loss of balance. These localized infections, while not immediately life-threatening like the septicemia of acute cholera, severely compromise the bird's health and well-being over a drawn-out period. Birds suffering from chronic fowl cholera often become emaciated and weak due to a poor appetite and the constant drain on their bodies from fighting the infection. They may be ostracized by healthier flockmates and can act as persistent reservoirs of shedding the bacteria into the environment and posing an ongoing risk to other susceptible birds. Managing chronic fowl cholera requires careful attention to isolate affected individuals and address their specific symptoms, though complete recovery can be difficult, and the long-term effects can be profound for the individual bird. Section 5. A feathered tapestry of susceptibility, birds at risk. Fowl cholera does not discriminate lightly. It casts a wide net across the avian world, affecting a diverse array of bird species. While it is perhaps most famously associated with domestic poultry, such as chickens and turkeys, its reach extends far into the wild. Many species of waterfowl, including ducks, geese and swans, are particularly susceptible. These birds often congregate in large numbers in wetland habitats, creating conditions where the disease can spread rapidly through contaminated water or close contact, sometimes leading to dramatic die-offs that capture public attention and concern conservationists. In the realm of domestic birds, turkeys are generally considered more susceptible to the severe, acute form of fowl cholera than chickens. Older chickens, however, tend to be more vulnerable than younger ones. Beyond these common farmyard inhabitants, other domesticated species like pheasants, quail and guinea fowl can also fall victim to the disease. The specific strain involved can influence the severity and outcome of the infection in different species, adding another layer of complexity to understanding the disease's impact across the feathered spectrum. Wild bird populations face unique challenges from fowl cholera. Outbreaks have been documented in various seabird colonies, shorebirds, and even some raptor species that might prey on infected birds. The stress of migration, breeding, or harsh environmental conditions can weaken birds' immune systems, making them more vulnerable to infection if they encounter the bacteria. For endangered or threatened bird species, an outbreak of fowl cholera can be a catastrophic event, pushing already fragile populations closer to extinction. The disease thus becomes a significant factor in wildlife conservation efforts. The susceptibility of such a wide range of birds highlights the ecological significance of. It underscores the interconnectedness of different avian populations, as the bacteria can potentially move between wild and domestic flocks. Understanding which species are most at risk and the environmental factors that might increase their vulnerability is crucial for developing targeted prevention and control strategies. This knowledge helps protect not only agricultural interests but also the rich biodiversity that our planet's bird life represents, a vibrant and essential part of our natural heritage. Section 6. Whispers on the wind, the spread of fowl cholera. The transmission from one unsuspecting bird to another occurs through several insidious pathways. The primary route is often through direct contact between infected birds and healthy ones. Bacteria are shed in discharges from the mouth, nose and eyes of sick birds, as well as in their droppings. When birds huddle together for warmth, preen each other or compete at feeding stations, these tiny pathogens can easily pass from one individual to the next, silently infiltrating new hosts and perpetuating the cycle of disease within a flock. Contaminated environments also play a crucial role in the spread of fowl cholera. The bacterium can survive for weeks, sometimes months, in damp soil, litter or water sources, especially if organic matter is present. Birds can become infected by ingesting contaminated food or water, or by inhaling aerosolized bacteria from disturbed litter or dust. This environmental persistence means that even after sick birds are removed, the threat can linger, waiting to infect new arrivals or susceptible individuals who were previously unexposed, making thorough cleaning and disinfection absolutely vital. Carrier birds are another significant factor in the relentless spread of this disease. These are individuals that have survived an infection, perhaps a chronic case, but continue to harbor and shed without necessarily showing obvious signs of illness themselves. Such birds can act as silent reservoirs, introducing the pathogen into healthy flocks or reigniting outbreaks long after an initial crisis seems to have passed. The movement of these carrier birds, whether through natural migration in wild populations or through trade in domestic settings, can transport the disease over vast distances. Furthermore, other animals and even inanimate objects can act as mechanical vectors. Rodents, such as rats and mice, can carry the bacteria on their bodies or in their droppings, contaminating feed stores and bird enclosures. Wild birds mixing with domestic flocks can introduce the pathogen. Even contaminated equipment, clothing or footwear of farm personnel can unwittingly transfer from one area to another. This multifaceted transmission network makes fowl cholera a particularly challenging disease to control, requiring a comprehensive approach to biosecurity and management to break these chains of infection. Section 7. Unmasking the hidden foe, diagnosis and detection. Identifying fowl cholera swiftly and accurately is paramount to managing an outbreak and minimizing its devastating impact. A presumptive diagnosis can often be made based on the history of the flock, the species affected, and the clinical signs observed, especially in cases of acute disease with sudden, high mortality. The characteristic rapid onset and specific symptoms like high fever, lethargy, and greenish diarrhea in poultry can strongly suggest fowl cholera. However, these signs can sometimes overlap with other avian diseases, necessitating further investigation to confirm the presence of. Definitive diagnosis relies on laboratory procedures. This begins with the careful collection of samples from sick or recently deceased birds. Veterinarians or trained animal health professionals will typically take swabs from the throat, nasal cavity, or affected tissues like swollen wattles. In cases of mortality, samples of internal organs such as the liver, spleen, heart, blood, or bone marrow are collected during a post-mortem examination, also known as a necropsy. These samples must be handled carefully and transported to a diagnostic laboratory under appropriate conditions to ensure the viability of the bacteria for testing. Once in the laboratory, several techniques are employed to identify. The most common method is bacterial culture. The collected samples are streaked onto special nutrient agar plates, which are then incubated under specific conditions that encourage the growth of the bacteria. If is present, characteristic colonies will grow on the plates, usually within 24 to 48 hours. These colonies can then be further examined microscopically and subjected to biochemical tests to confirm their identity as and distinguish them from other bacteria. More advanced molecular techniques, such as polymerase chain reaction (PCR), are also increasingly used for diagnosis. PCR tests can detect the specific genetic material directly from tissue samples, offering a faster and sometimes more sensitive method of detection, even if the bacteria are difficult to culture. Serotyping, which identifies the specific strain of, can also be important for epidemiological studies and vaccine selection. Accurate diagnosis is the cornerstone upon which effective control and prevention strategies are built, allowing for targeted interventions. Section 8. Fortifying the flock. Strategies for prevention. Preventing fowl cholera from taking hold is by far the most effective strategy for protecting avian populations, whether they are domestic flocks or cherished wild birds. Central to this endeavor is the implementation of robust biosecurity measures. Biosecurity encompasses a set of practices designed to prevent the introduction and spread of disease-causing organisms. For poultry operations, this means carefully controlling access to bird housing areas, limiting visitors, and ensuring that any personnel or equipment entering the premises are clean and disinfected. Such measures create a barrier against the unseen microbial world. Maintaining a clean and hygienic environment for the birds is another critical pillar of prevention. Regular removal of manure and soiled litter, thorough cleaning and disinfection of housing facilities between flocks, and providing clean food and water sources are essential. Can survive in contaminated environments, so reducing the organic load and using effective disinfectants helps to eliminate potential reservoirs of infection. Proper ventilation is also important as it reduces humidity and airborne pathogen concentrations, creating a less hospitable environment for the bacteria to thrive and spread amongst the birds. Vaccination offers another powerful tool in the preventative arsenal against fowl cholera, particularly for domestic poultry. Several types of vaccines are available, including inactivated bacterians, which are killed bacteria, and live attenuated vaccines, which are weakened live bacteria. Vaccination programs, when properly implemented, can significantly reduce the incidence and severity of the disease by stimulating the bird's immune systems to recognize and fight off infection. The choice of vaccine and the vaccination schedule often depend on local disease prevalence, the type of birds being raised, and the specific strains of bacteria present. Beyond these direct measures, stress reduction plays a subtle yet significant role in preventing fowl cholera. Birds that are stressed due to overcrowding, poor nutrition, extreme temperatures, or other concurrent diseases are more susceptible to infection. Providing adequate space, a balanced diet, and a comfortable environment helps to maintain a strong immune system, making birds better equipped to resist pathogens like. For wild bird populations, protecting their natural habitats and minimizing human disturbances contribute to their overall health and resilience against diseases like fowl cholera. Section 9. Turning the tide treatment and the future of avian health. When fowl cholera does breach preventative barriers and an outbreak occurs, prompt action is necessary to try and control its spread and treat affected birds. Though this can be challenging, especially with the acute form. Antibiotics are the primary medical intervention used to combat infections. Several types of antibiotics such as sulfonamides, tetracyclines and penicillins can be effective if administered early in the course of the disease. These medications are typically given to the entire flock through the drinking water or feed to ensure all potentially exposed birds receive treatment. However, reliance solely on antibiotics for control is not a sustainable long-term solution. The widespread use of antibiotics can lead to the development of antibiotic resistance in bacteria, making future treatments less effective. Therefore, antibiotic use should always be guided by veterinary advice and, where possible, based on sensitivity testing of the specific strain involved. Furthermore, treatment is often more successful in mitigating losses in the chronic form of the disease or in reducing the severity of an acute outbreak, if detected very early. For severely ill birds in acute cases, the prognosis often remains poor despite treatment. Beyond medication, supportive care can be beneficial for affected birds, particularly those with chronic infections. Ensuring easy access to fresh water and palatable food can help maintain their strength. Isolating sick birds from the rest of the flock is crucial to prevent further transmission. In some cases of localized swellings, such as in wattles, surgical drainage might be considered by a veterinarian to relieve pressure and remove infected material, although this carries its own risks and requires careful management to prevent spreading the bacteria. The focus must always include minimizing suffering for the afflicted individuals. Ultimately, the future of avian health in the face of fowl cholera rests on a multifaceted approach. This involves continued research into more effective vaccines, improved diagnostic tools, and a deeper understanding of the bacterium's ecology and virulence. It requires unwavering commitment to biosecurity and good husbandry practices in domestic settings, and diligent conservation efforts to protect wild bird populations and their habitats. Awareness among bird keepers, veterinarians, and the public about the signs of fowl cholera and the importance of prevention is paramount. By working together, we can strive to turn the tide against this silent threat and safeguard the remarkable diversity and vitality of the world's birds.